edition of a bioluminescent day, which was just an amazing experience, and that's where I really got passionate about learning about bioluminescence. So who here has learned about bioluminescence or seen it in person? Awesome. Yes. I'd say that's still one of the top moments of my whole life to see it for the first time. Yeah, so that definitely very true to what I'm doing now, which you may have heard about, which is the Microscope light wand, which is a eco-friendly glow stick that is powered with bioluminescence. So that's kind of what this whole talk will be centered around, and then after we'll get a chance to do a little bit of microbiology ourselves, and that will take place in the other room. So that's kind of going to be how this MakerMeetup goes tonight. Yes. All right, so design for rebirth is kind of what I've been thinking about for a long time, and it's how we can design the products we use and the life we live to be kind of beneficial for our planet instead of so drastically bad for it, which is kind of what we're dealing with right now. So there's a lot of amazing researchers that are open source, biomaterials, and really working towards building a better, kind of more open economy, as well as one that's kind of good for our planet and empowering. And that's a lot of just kind of what I've taken a lot of inspiration from while working on this project. A light wand, so you'll actually be able to see it. It's a prototype, so it's still definitely very much in development, but it does work. So I want to show you that too. And then introduction to your very own microbiome. So beyond the light wand, I think my big passion is talking about the microbiome. I kind of love it because it's invisible, but it's so important. I think maybe being a short person, I just identify with it or something. But yeah, it's just it's in us, it's around us, it's how we are connected to our environment. So you can kind of choose what kind of microbiome you want to look at today, but usually we make tongue prints to start, which is just kind of fun. Yeah, so now I'll focus more on the Microscope light wand. So yeah, it's the world's first eco-friendly glow stick. There's nothing ever really been made like this before. So when I talk about people wait, we haven't done that yet? Yeah, we need to do this. And actually, I've done now a lot of research on glow sticks, and there's over 500 million produced every single year. And they're non-recyclable, they're plastic filled with this toxic chemicals and glass usually, so they're not really possible to recycle. And yeah, they're found, in the ocean, on the ground, in our landfills. They're super fun, they look really awesome at night, but again, they're just one of those things that we don't need to use anymore, which is awesome. And then this is the part that I'll go more into a little bit more detail about in the further slides, but I wanted to design something, again, going back to design for rebirth, that would be healing for our environment, not just benign. And so in the last year of my degree, I was able to do an independent study looking into biochar, which is a component you can add to soils that is extremely beneficial for soil microbiome and actually helps boost the local microbes. You're not adding any kind of new species or new seeds or anything. You're working with what's already there and helping to bolster that. I'll talk about how later. Yes. So it's something that I want to kind of pass on to everyone, because I think you could really, apply this design principle to everything, to absolutely everything. And there's a lot of information now just online that you can kind of use to start to rethink kind of everything that we use. So design for rebirth, so it follows that life, death, and rebirth cycle that is kind of essential to life on this planet. Biomimicry, beauty, for function. So with the light wand as an example, it lights up, it's awesome, it's gorgeous, but then kind of the functional part is that at the end, when it degrades, it's then beneficial for the microbiome. It's how much uses biomimicry principles kind of in the utility of the light wand. And then from the ground up, we're sort of for the microbiome. So it's kind of the basis for life on this planet. It's primary producers, they degrade kind of everything. It all kind of cycles back down to that very base biotic level. Yeah, so we are still reliant on it. And right now, a lot of our agricultural practices and chemicals are destroying it. So, everything we can do to help it really goes a long way. And this is just a great way for me to now talk about it to all of you. Alright, yeah, so it's based on bioluminescence. And this is a question I get a lot, like how do all the little algae live in the white wand? How does that work? And it's not actually a living algae in the white wand. So it's recombinant protein. So it's based on this enzymatic reaction, which is a two-part system with luciferase, which is the enzyme, and luciferin, which is the molecule. They don't actually need to be in a living organism to do that reaction. The enzyme will actually do that on its own if the necessary cofactors are present. So that's what's happening in the white wand. The enzyme works when you add water, but you don't need actually to capture any living algae or anything. So that's how the bioluminescence works, and it's really a fascinating topic. I would love to talk more about that after. And this here is actually it's a folded kind of one way to look at a protein, and this was done on Primal, which is a protein engineering software. I just wanted to show you that this is kind of some of the structures that scientists work with when they're looking at proteins and how they work, how to make them better. You can add different kind of functions depending on the shape. So that's something that you might see come up in the future. And biomaterials. So if all this was encased in something plastic, it wouldn't be as good. So we're using a seaweed-based packaging kind of tubing that is clear enough to transmit light, but it's going to be biodegradable in terrestrial and aquatic ecosystems. So a lot of biomaterials are coming out now, which are amazing. They're not oil-based, but they're not actually biodegradable in the natural environment. And it's a step forward. I don't want to some biomaterials are bad because it's such a step forward. But with the light wand, I really wanted it to be beneficial when it's degraded, and I knew people would be throwing them on the ground. That's going to happen. So it's okay, how can we actually have that be a good thing instead of a bad thing if it's a behavior that's already happening? Sustainably sourced, so a lot of these materials are already kind of thrown away. So, for example, in Indonesia, there's a lot of seaweed farmers, but if it's not food grade, then there's not a big market for non-food grade seaweed. So there's this kind of alternative market now that's popping up to purchase that from the farmers, give a little bit of a secondary income source for the seaweed that was already harvested, but not graded for food grade. So that's just one type of economy model that I think is awesome. And there's a lot of examples like that. Yeah, it's actually edible. So yeah, I'm not going to encourage you to eat it, but that's just kind of the level of safety that we're looking at for the white wand. And here's the Biochar piece, which I love. Originally I did want to make the light wand a seed bomb. So I was looking at actually with Rebirth-a and Paul Sammy's company. They have a blend of restorative fungal and microbial species for the Pacific Northwest. There's this blend of over 20 different spores. I was we can add it to the light wands, then people throw them away, it'll be so great. But it's okay, what if people who aren't in the Pacific Northwest were to use these? I don't know if it actually is beneficial in every single ecosystem. So I kind of, was just rethinking that a bit. And so I looked into biochar. And who here has heard of biochar at all? Yeah? Yeah, okay. So this here is a scanning electron microscope image of biochar that I took as part of my studies. So this is biochar before it's been inoculated. And in the next slide I'll show what it looks like after it's been inoculated. But what this is it's pyrolyzed plant material. So when you pyrolyze something, you're actually burning it in a low oxygen environment. So instead of undergoing combustion, it's pyrolyzed. And so instead of generating CO2, the plant cellulose chains, which are usually in this chair structure, are transformed into a very stable aromatic ring. And these types of charcoals are actually hallmarks of traditional burn sites all around the world. And archaeologists use them to do carbon dating because they're so stable for thousands of years. So now biochar is actually being used for carbon sequestration in soils because not only does it increase nutrient retention, water absorption, and it helps microbiome, but it also is a way to sequester carbon and store it in the soil long term. So really awesome work is being done on biochars, and again, sustainable sourcing of plant material that isn't used for food, but is maybe just burned, like in forestry, often there's these huge burn piles. Those could be pyrolyzed and prevent a lot of carbon from entering the environment. And here it is. So as part of my research project, I inoculated, it was a whole plant study, and I used different types of biochar, I used different concentrations of biochar, and then after 6 months, I fixed them and scanned them. I wasn't sure if anything would show up because the fixing process for microscopy is really intense. I was like, is there even going to be anything that will show up after I do this? There was. I know it doesn't look like much, but it was probably one of the most exciting things I've ever seen. So you can see it kind of zooming in. Yeah, so here's where it kind of zooms in on. And then this little piece right here was so cool because what it actually is a formation, it's called like an extra polysaccharide matrix. But what it is it's a way for this bacterial colony to communicate with each other. They transport nutrients, electrons, all throughout this chain and in soils that are very healthy. This is what actually forms this like crust on top of the soil. So you can actually pick up this like whole crust that'll stick together, and this is a huge component of it, along with different types, so like mycorrhizal fungi, for example. But yeah, so seeing that was amazing. And what it really is it shows that the bacteria is going into more of a community structure. So from a single cell organism, and then it's actually able to start forming those microbial communities in the soil. Okay. Yeah, so I'll do the demo. So all of these components are part of the light wand, and yeah, I'll show it to you now. So we get the lights turned off and the doors closed. Were designed for outdoor festivals, so I usually spill a bit of water.
That's okay, we have big sizes.
Okay, good. So this is like the tubing, there's a bioplug which holds the biochar, and 2-part bioluminescence system is inside. And then this one's been used a little bit, because it's one of my few prototypes at the moment. And then this is the cap, which is just 3D printed right now. We're working to get it manufactured out of recyclable aluminum. And then it just kind of clicks in and blows. So I'll pour it with the light on so I can see a little better, and then we'll turn it off and you can pass it around. That would be great too. Thank you.
Big shelves. Just from the box. Oh, but she wants to run out.
So the great thing about using the enzyme system instead of an algae is that with the algae, there's kind of a diminishing return. So bioluminescent algae usually is activated by shear stress on the surface of the cell. So if you were to disturb the water, then it lights up. But after a while, it will stop being activated like that, and it will be light. So you won't see very much brightness. They require a lot of nutrients to stay alive. And then you won't see a longevity of glow. You'll see small flashes, and that would be kind of it. So it should actually be used instead of a glow stick. It wasn't really viable. And I also felt kind of weird about maybe having these, they're just single-cell organisms, but they're still alive. And that's a question I get a lot. So it is nice to say that this is, it's more of a biochemical system than a living, biotic system. And there's just amazing things being done in protein engineering right now that we can talk about after as well. Okay. So,
drum roll.
My hands always kind of shake when I'm doing these talks, which never goes well. Okay. So I'm going to pull the cap, and I'll shake it up a little bit. And then I'll turn the light off. So you can see it glow.
Wow.
So there it goes.
Yeah.
Here, I'll take it off and we can pass it around.
What is the life of the light?
Right now we're working with a few different systems. I think this is one of the quicker dissolving ones, but we have it up to about an hour. And we're working with a new system that will last even longer, but that's where a lot of the development will be in the future, is to get it up to the kind of grade of industrial strength light sticks. So here we can pass this around. And the kind of neat thing about it is that you do have to shake it to activate.
You can see that, so then when you're dancing,
Yeah,
it's just going to get a lot brighter.
exactly.
That's a good thing.
And I'm always kind of trying to see how long I can make it last in between, just like let it get quite still and then and then keep it going. Yeah.
Nice. That's amazing. Well, we just got water,
Yeah.
And so when you sold the kit, it would come with a cap.
Yeah. I'm thinking what we'll do is have the cap portion plus a kind of starter kit,
Yeah.
and then you can kind of get refills of the kind of longer tubing part and the actual bioluminescence. So this will be kind of a
I thought that was
thing.
just your necklace, so you could
Yeah.
like, if you got decorative stuff on the cap, like I wonder if you could make the cap such that afterwards people could wear it as a necklace.
Definitely, yeah. That's what we're working on, to make it look kind of nice enough people would like to wear it.
Yeah.
Yeah.
I thought it was a bullet.
Yeah. We might make it kind of darker so it doesn't look so cool.
This is the weight-based enclosure, this one as well?
That one is the it's not. It's
Okay.
the prototype version. Yeah. Yeah.
How long does the traditional glow stick last for?
It depends. So the ones you get at the dollar store are kind of 2-3 hours,
Okay.
and then there's ones. Actually, the biggest purchaser of glow sticks is actually military use.
Yeah.
And they use industrial strength versions that last kind of up to 18 hours.
Wow.
So we got a long road ahead of us.
Right.
Yeah.
But for festivals, like an hour would be enough.
Yeah, and
Maybe
those are
less.
definitely
Yeah.
kind of our early adopter market. But you'll pass it back there.
Yeah. Nice.
And so I think we're at the kind of question and answer part.
Hold it
too,
up for the
so-
camera.
Wow.
So,
Yeah.
one quick question, Paige. What's
Yeah.
the smallest volume that you can make and make it still glow?
You could we could really just add drops of water.
So you could even make earrings.
Yeah, you could make earrings.
Cool.
Definitely. Yeah. And like I mentioned before, I worked at Core Systems here, and they have this beautiful industrial grade like phosphorescent powder. It's like, oh, it'd be so nice to have a little bit of that in each of the caps, so that you can kind of like make that glow too. And those are used for glow-in-the-dark pathways. So actually, when I was doing my position in the bioluminescent bay, they were actually really struggling with light pollution around the bay, and that really impacts the dinoflagellates because they actually have this whole internal shift that happens every 12 hours. So during the day, they synthesize, and then at night they produce bioluminescence. So that's their whole internal cellular structure actually flips. And so when circadian rhythm is impacted, they won't bioluminesce as much because that cycle is disrupted. So what they ended up doing is they ripped out a lot of lights that were around the bay, and they moved the parking lot about half a kilometer from this bioluminescent bay, which is a huge draw for tourists. It's kind of their main thing now. And so now when you arrive, you park your car, turn off the lights, you walk slowly along this path that is lit up with these glowstones, and your eyes adjust, and it's kind of magic. And then you arrive out to this bioluminescent bay, get a kayak, and you'll kayak around in bioluminescence.
Where is this?
It's in Vieques, Puerto Rico. Yeah, and they've been really impacted since the hurricane. I'm still quite connected with them, so I like to kind of bump it up. It's really one of the most beautiful places I've ever been. And they do amazing conservation work there, not only for the bioluminescent bay, but for a lot of sea turtles as well, and that kind of have their home on the island. Yeah, it's just a little island off the coast of Puerto Rico. It's like the world record book for the brightest bioluminescent bay in the world. Yeah.
So it doesn't even come close to real life.
here it's
No.
maybe nothing, or little green
Like,
ones.
well, every now
or-
and then when we get a flush, it's amazing, but it's like that there all year round. It's kind of the big thing there. Yeah, yeah. And when it rains, because like we're not allowed to swim in it anymore because for a lot of people we're swimming in it, but you know, we wearing lotions and sunscreens, so now that's stopped, which is one of the work that this conservation trust did, and now we kayak in it. But if you're lucky, you'll be able to go when it rains, because when it rains, the whole surface of the bay starts to glow, because that's when all of the bioluminescence is activated. Yeah, so if it looks like it's raining, you're actually in for an amazing night.
So when you're kayaking, when you're doing that, it's gonna light up.
Yes.
When you're
Yeah,
paddling,
on your
it's
paddles.
and then also like when you're seeing like the kayak breaking.
Yeah, and you'll see all these little fish. Like
And
the
the
kind
fish
of
are
outlines of the
too.
fish. Yeah.
That sounds so magical.
Yeah, definitely.
And it's the best in the world, right?
Yeah.
The world record.
Yeah, they got the world record. Like, I want to visit all the bioluminescent ecosystems, you know, one day. There's like termite mounds in South America. There's like the glowworm caves in New Zealand.
Awesome. Glowworm caves.
Yeah. So I've really been focused on, you know, algae. He's so neat. And there's been some amazing, David Attenborough has a documentary you can access online about bioluminescence. It's an hour and a half long and it's awesome. And they go into bioluminescent mushrooms and also one of the kind of curiosities that I'm not sure of yet, because scientists always thought that bioluminescence evolved for communication or potentially just for like a kind of side reaction as like a reduction of oxidative stress, because that's kind of the type of reaction that it is. But some mushrooms actually still bioluminesce in their mycelium. So not even in the fruiting body where you know it's visible, but actually underground. You're like, why does it do that? Yeah. So there's a lot we still have to figure out about bioluminescence and its functions. And that's actually one of the problems. So I don't know if anyone saw there's a Kickstarter for bioluminescent trees and plants a few years ago. And I heard that. But you know, they had some really good technology, but the project ended up failing because bioluminescence is a pretty energetically intensive process. And so animals and you know, species that haven't evolved to kind of handle that suffer because it's so intensive. And so they just couldn't get these plants to really like survive well when they were producing bioluminescence, which they kind of added into their genome. But it was such a kind of energetic load that it just wasn't kind of very well handled. And that's actually something that's a huge kind of, it's a challenge in genetic engineering and in biology work, where if you want to add something in, it usually, like in a few generations, just be kicked out because these systems have been developed, you know, with everything for so long.
Is this the new GMO?
So we'd be messing with something works and great for the market out when they're messing with the genes and these freakish things.
There's some freakish things going on in the world for sure. This isn't a GMO because it's not an organism. So GMO usually would mean like a living organism which can go on to reproduce and everything. So this is it's called a recombinant protein, which means that it's being grown up usually in expression system. So this bioluminescent protein, for example, is grown up using yeast as the expression system. So yeast has been used for decades now to produce enzymes that are used in like laundry detergents, insulin, and now also some bioluminescence. And it's grown up in a huge bioreactor, and then it is kind of purified out. And that's how most recombinant proteins are made, actually, is using yeast as the expression system because it just loves to grow. Yeah.
I was just saying, you can grow cannabis or like cannabinoids and stuff with yeast.
There's some amazing work being done there now. Yeah.
Like the yeast could probably produce everything, like a 3D printer of nature. Yeast is like, we can do everything. We'll make it all. It's just our job. That's what we do, right?
Yeah, I'll send you a few like Facebook groups that I'm part of that are really kind of targeted about kind of advancements in biotechnology for the production of like therapeutics, for different types of biochemicals. Did anyone hear last year about the kind of, they call it like a mutant enzyme that can break down plastic
Yeah.
40 times better than like anything else.
Mhm.
So that was produced in a lab, and now they're looking at that for hopefully the reduction of plastic waste and the ingestion of it.
In landfills.
Yeah,
All
yeah,
landfills, right?
The
yeah.
hard part is though, like, what about the domino effects of that we can't foresee? Like, when cars were brought out, they thought that was a great environmental thing because it was solving
Yeah.
the pollution problem of the day, which was horse poop. And so cars
Yeah,
didn't
yeah.
create any poop, so they thought, this is great, this thing's... And
Yeah,
now down the road, we know that cars had a lot of
Yeah.
other problems you couldn't see that
definitely.
have impacted us in huge ways. So
Mhm.
I always worry, like, okay, these things eat plastic, but what else? What are they creating, or what ripple effect will that
Yeah.
have that we don't know about?
Well, that's a huge question in synthetic biology, and it's really interesting the regulations around releasing modified organisms in the wild.
Yeah.
There are huge regulations around that, but somehow modern agriculture they sidestepped that before anyone else way back kind of in the 80s, and that's how like Monsanto, for example, was able to really to produce and then patent.
Genes and things.
Yeah, and so it's interesting now that kind of regulations have started to catch up to some of this technology. And I know when I talk about biotechnology, the first thing people think of is Monsanto
Yeah.
and Roundup Ready crops and pesticides and big agriculture. And I get it, like they've done horrible things, and it is really scary. And like in Europe now, they're doing massive bans. And like, I have some friends who are really involved in the Community Biology Initiative, which is kind of a worldwide group of scientists and activists who are really kind of like passionate about some of the potentials of biotechnology. But a lot of people in Europe actually are not able to do a lot of work that they would like to do because of the regulations, for better or for worse. Yeah. And one of the huge critiques on this kind of whole science is exactly what you said, that it is an experiment.
Yeah.
Yeah. So like a worldwide experiment is one thing besides doing something like small in the lab. But that's usually for things that are, they said, like modified to survive in the wild. And I know, like, I was working in a lab at UVic, and they were actually not designing things to survive in the wild. They were looking at ways to turn things off. So providing like an environmental off switch, whether that was through temperature or through different types of media, or in one way, let's say for the plastics, you could engineer a species that would only survive when eating a specific type of food source, like let's say a certain type of plastic. And then when that food source was no longer available, they couldn't survive and they would die. It's just kind of one of the many options. But bacteria evolve so fast, so it's kind of one of those things. One thing that I will just kind of say though about kind of this lifelong project is that like it's not a genetically modified organism. There's nothing that is being introduced that has never been made before. It doesn't go on to reproduce. Yeah, it fully biodegrades, so it's not. So that's kind of the distinction that I really like to be clear on, because I so hear concerns on biotechnology.
Are you the only one looking at a sustainable light wand? Or do you have competitors out there?
Currently, no, but I'm sure they're coming.
Oh yeah, we saw you doing the
So we'll see what happens. Like I know there's people out there who have way more resources than I do. I started this as a student project. I reached out to the this bioluminescent research lab who initially started studying it for cancer imaging. They never thought about it as like a light source in terms of chemiluminescence, and so it's really like it's been a long-term project to get it to this point, and I've really done it all through like so many emails and lots of testing, and just kind of I don't really care about it. But we'll see what happens.
Are you going to patent your
your
formula so that people can't?
Everything is patent protected at the moment, which is also why I do feel totally comfortable talking about it.
They can find their own way to do it, but they can't use your way and profit from that.
So there's pros and cons to that, So I know I'm really big on the open source movement. So I was looking at how to do this. It's like, do I actually patent this? but then I also want this to happen. So I was like, well, maybe I need to. And then I did end up deciding to patent, which was this whole process, but I learned a lot about how patents work. And actually, one of the biggest things I didn't even think about with patents is, because at the beginning I wasn't really sure if I wanted to, but then it turned out that even if you put into the world that you were working on this project, but you didn't have the kind of legal patent behind it, then someone else could patent it and then literally like sue you for doing your own idea. And so it's just kind of a weird system where you're almost locked into needing to do that. That was, I was like, what?
Well, I mean, the thing with that is that you can team up with people who have resources and, and take it to larger scale.
Definitely.
Other people want to do it, they can join you. You're
Yeah.
not like, oh, it's just mine, no one else can think. It's just, you don't want people stealing ideas.
Yeah. And I just have one more thing to say about that is we have, I now do have a small team. I have an amazing co-founder who is much more kind of business development side. And also we've been kind of early accepted into Rebirth-a, which is a life sciences accelerator program. So it's kind of happening, yeah. Thank you.
The dragon's dead there. We need it for the Zoom.
Just a question from the internet. What is the shelf life?
So that's something that we still need to test with our prototypes because, as you saw, they're activated with water. So if they're exposed to any kind of moisture, then that will rapidly degrade them. So right now they're stored in like a very sealed container, but unfortunately that doesn't quite fit in with all of our kind of biomaterials work. So that's something that we're still looking at. Like the shelf life is very long if they're kept in a very dry, stable environment. That isn't exactly the back of someone's backpack while you're going to a music festival. So we're going to be doing a lot of user testing to figure out kind of that kind of information. But thank you for asking. Cool. Question would be to do that. So I would love to transition into doing the kind of hands-on activity. Is everyone okay with that? Any last-minute questions? So,
Kind of related, but it's like bioluminescence. Is it usually in the visible spectrum, or does it go like into ultraviolet and infrared?
Gosh, that's a great question. And I've only heard of bioluminescence that is in the visible spectrum. And there's like kind of protein, kind of escapades. I don't know what to call it. Really, there's explorers who go searching for specific types of bioluminescence, and especially actually fluorescence. So for brain imaging, you want a wavelength of light that can pass through the material in your brain, but then also will fluoresce at a way that is able to be picked up by a medical imaging device. So we actually found a fluorescent protein that will be excited with red light, will emit in blue light. And that's
so you can see it
super useful, for example.
Cool.
So what's the difference between luminescence and fluorescence?
Okay, yes. So, there's so many different terms. That's luminescence, that's the end word. So fluorescence, that would be like a car, headlight, or not even like that. It's like when you shine the light on it, it immediately sends light back. So it's instantaneous, but if you were to remove the light source, it would no longer create light. So it's more reflective than anything, but usually with fluorescence, it's not like truly reflective because there will be a kind of energy shift. So usually it's shifted to a lower wavelength is kind of how fluorescence works. Phosphorescence is things that glow. So glow in the dark stars, glow in the dark pathways like I was talking about. They usually have a different chemical structure. So light is actually absorbed and then re-released. And that process is kind of what generates these long glow-in-the-dark states. And then bioluminescence, which is kind of its own thing, is actually the chemical production of light with a chemical kind of source. Or it's the breaking of the chemical bond that produces the light in this case. It isn't actually absorbing photons directly. Is the difference there.
So phosphorescence in the ocean are not bioluminescence.
Well, they are. I think they're just called the same thing. So in the ocean, that's absolutely like bioluminescence, like living light.
Yeah.
They're creating it from a molecule that's found in most ocean water.
Yeah.
Yeah.
Okay.
People call it phosphorescence,
Yeah.
and it's, I think it's just kind of become a colloquial term.
Yeah.
But it's technically, yeah, technically phosphorescence is like that kind of long extending flow. Yeah, I could go on about that too. It's really cool how it works because you get this, like in the crystal structure of this molecule, you have these dopants, which are really heavy, usually heavy earth metals, and then there's this crystal lattice structure. And when the like photons hit, the like electrons are kind of excited to this heavy state, and then usually they're re-released. But because of this crystal structure and this like really kind of like heavy earth elements, they're caught in what's called like a metastable state. So where they won't actually be re-released right away, and they'll kind of be like held there and slowly re-released. So that's the long-lasting glow.
And can you get like different colors of glow sticks too, or is there just the one color?
Right now we're working on green as well, which I would love.
Awesome.
Yeah. Like really there's so many options for that. Basically as many colors as like we can see, and maybe even more than we can't see. But this is kind of what's initially been developed.
So you've seen colors that we can't see.
What is it like a mantis shrimp? Like I can see like 18 different colors. It's amazing. But I was like, oh, it'd be neat if you could have something that, you know, maybe produce light that then was absorbed to produce, you know, some other kind of light, but you couldn't see the first one. Then to get these kind of like chains happening to produce the end light product, but it's probably going to take a lot of energy and a lot of time, and yeah, probably isn't the most efficient system, but yeah. I just looked
How
at
did you first get interested in this?
It started way back. Like, I don't know. I've been an environmentalist kind of person since like I was very young. So you know, like the Biochar marmots that are here.
Yes.
I was like, really hardcore about saving them when I was in elementary school, and I did a lot of work for them. And then I ended up being able to go to this like United Nations Children's Conference on the Environment when I was in grade 5,
Wow.
and that was huge for me because there was people there, you know, all ages doing brilliant work around the world, and it was actually helping predict that year. So I went with my grandpa, who was like also just a huge environmentalist from Saskatchewan. Yeah, yeah. So that's kind of, I think, like where I would say like my drive comes from, absolutely. And then, you know, I love going to festivals. I absolutely just love it. I find the community to be amazing. I just like have a lot of fun dancing. And actually I want to talk about this. Like this is one of the things that I think a lot of people struggle with in the environmental movements and activist movements, where it's so depressing and there's so much like really hard work that takes so long to have anything happen. It's so draining, and I felt that a lot, like in my environmental studies classes, you know, doing political ecology, and learning about all the ways like our government is corrupt, and like how our laws have like embedded this kind of horrible like system, and it's just a lot for like one person to absorb and like take responsibility for. And that's something I know I struggled a lot with, and so going to festivals I actually found like to be this like really positive, like beautiful experience where I connected with people. It's just like feeling joy about like being alive, which so healthy. And like truly our earth is like this amazing, amazing place, you know, and like I get so stuck sometimes feeling like how can we like fix it, how can we like do something to help, but then when I have those moments of like appreciation, it really helps keep me going. Yeah, and then so I would go to these festivals, but I would see all this garbage too. It's like, oh no, like it's here too. And I come home feeling so happy, but then it was such a contrast. And there's actually some amazing movements now that I've heard of that are creating greener festivals. And at that point I was really, really like kind of obsessed with bioluminescence, which is getting better all the time. And so when I saw glow sticks kind of all over being used, it's like, maybe we could do this, Yeah. And so I really started working on it, yeah.
That's gonna like inspire other people in other facets of things that come to festivals.
Hell
Like
yeah.
making more packaging that's like you're saying, beneficial.
Yeah.
So when you're throwing your things down, like.
Like, I just attended Otherworld like just this past weekend, and it's a Leave No Trace event that happens on the island. And I always get around these festival missions when I'm at festivals. I carry like a pocket of tricks basically, and like wander around like, hey, you're on a festival mission. And one of them is like stop and pick up the closest 5 pieces of garbage that you see, and put them where they belong. And there literally was no garbage on the ground there because everyone was so self-responsible. It was beautiful, and I've never been to anything like that. And it really made me so hopeful. Everyone there really cared, And so yeah, just being around kind of 1,000 of those people was just so hopeful. Yeah. And everyone I met was doing really legit work, on the outside. But and also was looking for ways to just kind of have mentally healthy experiences as well, because it can be a lot. Yeah. Yeah. So thank you. Okay. Yeah, I would love to get you all now to because microbiome is kind of what I love talking about the most. And we have a hands-on activity, and I have a couple slides about it actually before we go to the other room. So I kind of mentioned in part of this DIY bio movement, and that's kind of, science can be so inaccessible. I've had some health challenges throughout while I was in school, and I found that the microbiology and science programs were very intense, and if you couldn't give it 120%, even if you were physically unable, then you were looked down on, and you suffered because of that, even just in how people saw you. So I've been struggling with my own life, and there's so many different ways that you could struggle within an academic community, so many ways. And so that's kind of why I started working towards community bio and DIY bio, biohacking, there's a few different names, but and there's community bio labs for citizen science projects. And one of the things I'm really excited about is kind of environmental quality monitoring and just doing kind of simple self-tests, whether it's for your health, whether it's for the products you use, whether it's for environmental samples that you have, all that can be done very safely and very simply in community bio labs. You just need a few instruments, maybe a few pipettes, a microscope if you're lucky, but not even necessary. What we're doing today, I ordered agar off of Amazon, a few petri dishes. You don't even really need those. You can literally use cupcake cups if you want, because they come very clean. And your agar, a little bit of sugar, and then beef bouillon cubes for nutrients. And that's a really great kind of general nutrient agar mix for growing up quite a few different species of bacteria, for example. So that's what we'll be doing today. We'll be actually making that agar mix. We'll be doing that in the other room. I'll show you how it's all done. You can all pour your own petri dish, and I'll talk to you a little bit about sterile technique and how to kind of keep your area clean, because what we want to grow will be only from the sample we choose and not from the air or the table or our hands kind of thing. And we'll also talk a little bit about how to design and experiment with controls. So what we'll do, you'll all have a couple of dishes for yourselves, but we'll also have one dish that just sits open on the table just to see what else will drop into it. I guarantee there'll be a lot. Yeah, wherever you go, there'll be a lot. And then we'll also have them closed. Then if you were to do your own experiment, you would actually just want even just one close for yourself, just from the pouring, because that way you can tell that whatever grows isn't from your starting mixture, that it's from somewhere else. So you can kind of develop controls at every step of your experiment so that you can verify your results along the way. So we'll be doing a little bit of that. Julian has set up a little, what do you call it? It's not an incubator.
Oh, it's an environment chamber, but basically it's a controlled oven. So
It's
we
a
got it all
controlled
set up.
oven, but we can set it for a specific temperature to grow up these bacteria in, and then we'll be looking at them. Usually it takes at least 24 hours because we're not concentrating the bacteria beforehand. It'll probably take a little longer to see any big colonies. We'll be sending photos, and you can kind of get a little info on your own microbiome from this little experiment. So
So I just drank kombucha, so there's gonna be like
That'll be interesting.
kombucha bacteria is
Okay,
going
what we
in
can do for that, pour a little kombucha on one plate and then lick the other, and then ones that grow on just your plate that you lick will probably likely be what's there already compared to the like, kombucha plate, you know, that kind of thing. Yeah, okay, let's do it.
I think I like a
Yeah.
little drip in it at least.
Oh, sorry, I was going off script. This is actually a slide that shows the different microbial species that are present like all over the human body.
Oh, cool.
And the diversity is just amazing.
There isn't any way you could send that, like, to
Yes,
people
we'll be
who
posting
hear
this.
you post
Yeah,
it?
yeah,
Cool.
yeah, for sure,
That's something
for sure.
I want to know about.
And so like this idea of like the holobiome, which is just like we are just one species of many that forms like
Yeah.
this whole
Are-
thing.
There's so much bacteria living on us at any given time, and it is
And
fascinating.
there's some really interesting studies around like your gut and the soil and the interconnections, and it like a big concept is just connection to the land, and if we like strengthen our connection to the land, that will really help with the environmental movement and not only our own health but like help our communities. And I feel like the gut and soil microbiomes are kind of the like physical connection to the land and that reciprocal relationship that we have. So, okay, they say reciprocal, I just want to ask a question. So, if we are like eating food from the like ground, and that becomes like, what is the reciprocity that happens? Yes. Yes. You get a gold star. Yeah.
But some of our food
but we
goes
treat it
down
all.
in the ocean.
Yeah.
It does
Yeah.
not in the ground.
So that like reciprocal like relationship, it's really been like cut up, you know. So that's just something I want to like drop it on your heads for right now.
So don't people use night soil?
Yeah.
Like in a lot of countries, people still use their poop to put back in the earth.
Yeah, and there are pros and cons
Yeah.
to doing that or not. I actually know in town you can get like it's called like rocket fertilizer, and that actually is like a human waste management.
Really?
Yeah, so people are doing some interesting work for sure.
Branding for the win.
Yeah, and I'm curious what we'll be doing, the microbiome mapping. So you really, like you could do like every part of your body, that would be like, I want to do that as kind of like an art, like kind of piece where, you have like people stand and then on each part there would be like a picture of their like agar plate that comes out, for example. And then when I will be taking photos of these agar plates after, so if everyone is okay that I use them for maybe like an art project or post them as part of this, like let me know, and if you're not okay, let me know, and I will write it down and remember, because because it's just personal information. Yeah, and I won't use any names or anything. Yeah. And here's a little art piece I made that was from my like face skin, and then I just like put a face behind and took a photo. So yeah, thank you so much.
Wow, this is so good.
Yeah, let's go to the room.
Okay, so just 1 second. 1 second.
Okay.
That's okay. Just 1 second, just because we have hello internet. So we're going to close up for now, but I just want to say thank you so much, Paige, for getting in touch with us and saying, oh, I'll do a talk. I think it's been a great way of restarting up the Maker Meetups. And so if you found this interesting, a couple of things, let your friends know, because we plan to do something like this once a month, a little bit of a break over summer. But we've got people talking about ham radio, we've got people talking about some other art stuff. Anyone you know who might do something interesting and be willing to come and show, that's what this is all about. A chance to learn and actually do. And we will plan to do it basically on a monthly basis. And again, Paige, thank you so much. And we will be doing another session a little bit more in depth on July 6th.
Yes.
Details to be determined, but I think Paige has got some super cool ideas that we might be able to do. And if you're interested in this, part of a makerspace is doing things that you are interested in. So if you find this DIY biology of interest, get in touch, help me make it happen. There's nothing stopping us doing some DIY biology right here. We've got an expert we can lean on for some information. She doesn't live in the valley all the time, but she can guide us.
Yeah.
So if you're interested, get in touch and we'll make something. So thank you everyone. Liam, can you push the off the air button?